um, uh, have a look again at the cell membrane. Now, I've referred to the cell membranes often so far. And so you'll gather from that of its singular importance in the structure of the cell, but also in the functions of the cell. So we need to spend some time looking at it in more detail. You will remember from biology that the cell membrane, that membranes, whether it's the plasma membrane or any other membrane, consists of a skeleton which is built out of phospholipid. Now, the phospholipids are a kind of a schizophrenic molecule because they consist of one portion, which is these long hydrocarbon chains, which are called fatty acids. They're actually lipid there. This is an oil. And just like any oil, it is highly hydrophobic. It is repelled by and repels water and does not dissolve in it. And you know this because if you pour oil onto water, it floats, and it floats for a reason. It floats because it's trying to get away from the water. So this is portion of the molecule is hydrophobic. This portion here, however, is highly hydrophilic. It dissolves in water very, very easily. This part here consists of a phosphate group here, which is joining other chemical groups here, and a molecule here called glycerol. Glycerol is an alcohol sugar, also highly, highly soluble. So this whole unit here, we kind of think of it as being the phosphate head of the molecule, is highly soluble. This here is highly insoluble. So this molecule is forced now, whatever environment it's in, it has to behave in a way which satisfies those needs of the different parts of the molecule. It satisfies the characteristics of those two parts. So the way I want you to imagine it is, first of all, if I was to just take, this is liquid, by the way. So if I was to take a small drop of this and drop it onto the surface of a beaker of water, how would it behave? There's only one way for it to behave, and that is to form a film on the surface of the water with these heads dissolved in the water and the tails sticking up out of the water, away from the water. And that is exactly what it would do. Now imagine that I force it underwater. I absolutely force it underwater. Maybe I take the vessel and I shake it like this and force the film to break up and go beneath the surface of the water. What will it do? How will it behave? Initially, it will start to form spheres where the heads of the phosphate heads are on the outside, forming a shell dissolved in the water, and all of the tails will point inwards and exclude all the water. So that's fine. Now we have these little spheres. Now I'm going to be really cruel to them, and I'm going to shake them up so vigorously that I force water into the center of that sphere. Do you see what I mean? I force water into the center of that sphere. Now, how does it behave? There's only one way for it to behave, and that is for it to form a double membrane. And this is what we see here. You can imagine if I did that, water on the outside, water on the inside, then the heads dissolved in the water on the inside, heads dissolved in the water on the outside, and all the lipid tails facing inwards, excluding water, excluding all the water. This is the basic structure of the membrane, of the plasma membrane. It consists of two leaves of phospholipids, and they are oriented so that they satisfy the requirements of their respective relationship to water. Highly insoluble on the inside, highly soluble on the outside. And this is the basic structure of the plasma membrane. And here is an electron micrograph. You can see here on this light band in here, this is all the fatty acid tails. And this stained darkly on the outside of phosphate or glycerophosphate. This glycerophosphate because it's glycerol and phosphate groups. All of the heads, soluble heads on the outside. So that is the skeleton of the plasma membrane. It's not the whole plasma membrane, but that is the basic skeleton, the basic structure. It's formed from these molecules. Now, the membrane is a living thing. It has living function, but the living function is not conferred by this phospholipid bilayer. Instead, it's conferred by proteins which are embedded in the phospholipid bilayer. The first thing to realize is, I've said it a couple of times, but may not have struck you strongly. This is actually liquid. It is held together by the forces, by the hydrophobic forces of the water. But it's also held together by molecular attractions between the molecules. And even though those attractions are relatively weak, there are so many molecules that it has a certain strength. It has a tensile strength to it. But it is a liquid. And so the model that I'm going to show you of how the membrane is structured and how it works is called the fluid mosaic model, because it truly is a fluid. And the proteins that we're going to talk about are actually dissolved in that fluid. So have a look, have a look here, and we start to get closer to what the membrane actually looks like in real life. And that is, here's our the skeleton, the phospholipid bilayer here. But embedded in and associated with this phospholipid bilayer, there are many, many different kinds of protein. Some of them span the whole membrane like this. They go from the outside to the inside. And those are called the integral proteins because they're integral to the membrane. But there are lots of others as well, which are associated with one or the other side of the membrane. And those are called the peripheral proteins. Now, these are all very, very active. Most of them are proteinaceous enzymes, and they are doing many different things. They enable, for example, in bacteria, most of the really important biological functions take place at the membrane because that is where the proteins are. That is where the proteins which drive these processes are located. So, for example, the processes of respiration in bacteria take place at the membrane. Cell wall synthesis takes place at the membrane. DNA replication starts at the membrane. Very importantly, remembering bacteria have no eyes, they have no ears, yet they need to know what is happening in their environment. Many of these proteins are sensory, and they will sense what is happening on the outside, for example. And by reacting with what is happening, maybe they're measuring the pH. The protein sees that the pH is getting out of range, out of safe range. It may spark reactions inside the cell to protect the cell. If it is sensing that, for example, oxygen is getting low, it may trigger fermentation processes on the inside. Many of these proteins are sensing what is happening and relaying information to the cytoplasm for certain processes to begin. But one of the most common and important functions of these proteins is to regulate transport of molecules to and from the cell, regulate passage of molecules into the cell, and regulate the passage of molecules out of the cell as well. And there are two, this we refer to as transport, as molecular transport. And there are two ways in which this is done. The one requires no energy input from the cell, and that is called passive or facilitated diffusion. The other requires an input of energy from the cell. It has to tap its energy reserves to drive the process. And this is active transport, and that utilizes ATP. So we'll get on to some of the other functions of how this is done. But for the moment, just be satisfied. We have that structure, the fluid mosaic model, the phospholipid skeleton with its associated proteins. Just have that in mind. Okay, so if we move out from the cytoplasm, then we've got our cell membrane. Cell membrane ensuring that conditions outside the cell can be different from those inside. But then on the outside of the cell membrane, we have the cell wall. The cell wall is completely different structure to the cell membrane. It has no transport facilities at all. It is purely a molecular reinforcement mechanism, put it that way. That it does regulate the cell shape, and it does provide some mechanical protection from crushing, or it guards that fragile lipid bilayer that is just underneath it. But that's not its main function. The main function of the cell wall is actually to protect from osmotic damage. And I've described to you carefully beforehand how the cell will swell under osmotic pressure and up against the cell wall until the pressure inside the cell, the turgor pressure inside the cell, equals the osmotic pressure trying to push water in. And when that happens, the increase in amount of water inside the cell stops, and a static, stable condition is reached. So the cell wall is extremely important, but it also provides us with some really important clinical information. Now, the cell wall itself can be antigenic. In a disease, in a pathological situation, in a disease situation, the immune system can respond to certain components of the cell wall. So we say it is antigenic. But it's also a target for antibiotic strategies. And I'll explain to you the way in which we use antibiotics in a second. But I point out that there is a natural system which acts against the cell wall, especially of this group that we're going to talk about that have a very thick, heavy cell wall. And this is a substance called lysozyme. Lysozyme is very widespread in the biology, but especially in the mammals. We have lysozyme, for example, in tears, quite common, and in mucous membranes all over the place. And it's there to act against bacteria. And what it does is it actually breaks down the cell wall. And once it breaks it down, all that osmotic protection is lost, and the cell will burst under osmotic pressure. So the antibiotics like penicillin also act against the cell wall, but in particular against one group. And these are the gram-positive bacteria. And the gram-positive bacteria have a very, very thick cell wall. Gram-negative bacteria also have a cell wall, but it is not thick. So in both cases, the cell wall is made of peptidoglycan, which is a very complex carbohydrate molecule. I don't expect you to remember all of these names. All I want you to remember is it's made up of rigid fibers composed of the polysaccharides. And then it is these fibers are cross-linked with peptide chains, proteinaceous chains, which link them all together. So it begins to form a rigid basket structure. In the gram-positive bacteria, there are many of these layers laid on top of one another. Let me just jump here. So here's our cell membrane there, plasma membrane. And here on the outside of that is this thick layer of peptidoglycan. Here you can see the different leaves of fibers laid on top of one another, but these are bound together. First of all, they're bound horizontally by peptide bridges, and then vertically here, they are bound by teichoic acid. And teichoic acid is a very, very strong binding agent, which ensures that this is a rigid entity, and it becomes extremely strong. So this type of peptidoglycan is particularly susceptible to antibiotics like penicillin. Penicillin especially acts against gram-positive bacteria. So here's the typical gram-positive structure, just the normal cell membrane, skeleton with all its embedded proteins, and here on top, thick layer of that peptidoglycan. Just let me check. Oh yes, I should say that this is the teichoic acid, which ties the whole thing together and to the membrane. This teichoic acid is actually clinically significant because it is strongly antigenic, and it excites a reaction from the immune system. Okay, so that's the gram-positive, relatively easy to understand. It's quite a simple structure. The gram-negative, however, is much more complex. This division between gram-negative and gram-positive took place very, very early in the evolution of the prokaryotes. And we can see traces of these in the fossil record, for example. So the gram-negative consists actually of two distinct layers outside of the cell membrane. So here's our cell membrane. I'll go to a bigger one in a second. Here we are. Here's our cell membrane there. As before, the phospholipid structures, and then the proteins embedded in that. Here, however, is the peptidoglycan layer there, and it is extremely thin, usually only one layer, some maybe two, but still always extremely thin, and there is no teichoic acid stabilizing and binding it either. All it does have peptide cross bridges, but it has no teichoic acid, which is what really confers the strength to the gram-positive. But outside of the cell, this then is the cell wall itself. Outside of the cell wall, there is this structure here, which is called the outer membrane. And the outer membrane, at first glance, looks very much like the cell membrane. And it certainly is, it's a bilayer. And its bilayer structure is conferred by the fact that it has one part of the constituent molecules are hydrophilic, as before, these heads. And then it has hydrophobic tails, which face inwards. But in fact, this leaf here is a mixture of two kinds of molecule with that dual nature. The first are our typical phospholipids. They are with the yellow heads here. But these ones here with the red, shown here with red heads, stained red here, are called lipopolysaccharides. And they are somewhat different in structure. And they are clinically extremely significant. I'll return to that in a minute. just be aware that this this is called the outer membrane. And just be aware that it has those two kinds of molecules. It has phospholipids and lipopolysaccharides mixed in it. Now, I just want to take you back to this. I want you, if you have printed these out, I want you just to make a correction. Here, this sentence reads, the outer layer is of the outer membrane is made up of phospholipid and in your notes it says is made up of lipopolysaccharide. It's not entirely made up of lipopolysaccharide. It is a mixture of phospholipid and lipopolysaccharide. That's important, so please add that in there. Okay, so this sets up a situation where we have lots of nuance and lots of detail we need to see. The first is that between the outer membrane, this whole structure here, and this here, which is the cell membrane, there is a space. And that is called the periplasmic space. And it is filled with a fluid which is extremely packed with molecules so that it is like a gel-like substance. And that is called the periplasm. So the periplasm occupies the periplasmic space. Inside the periplasmic space, this is the cell wall here. And spanning the outer membrane, there are many, many, many proteins. It also has associated peripheral proteins as well. Many of these are actually transport proteins. They govern the passage of transport of molecules into the periplasm. And then the membrane itself is responsible for transport of molecules from the periplasm into the cell or the other way around. This periplasm, it turns out, is like a storeroom. And gram-negative bacteria have inside that periplasm, they have solutions, digestive enzymes, of rare nutrients, of all sorts of things that they store inside the periplasm, ready to pass the digestive enzymes out, but nutrients and things like that into the cell. So this is a very active, dynamic structure. This whole structure here outside of the cell membrane. And we need to also, because it is clinically extremely important, we need to have a look at these lipopolysaccharides again. The lipopolysaccharides actually are clinically significant because they have some portions of them which are highly antigenic. The lipid part of the lipopolysaccharide is called lipid A. And lipid A is very highly antigenic. It is an extremely dangerous substance. And if it is liberated, for example, in human beings or mammals, if lipid A is liberated into the bloodstream, it can induce such a strong immune response that it will result in shock. Now, that's not shock like, it's shock, clinical shock, which is a catastrophic collapse of blood pressure. It's part of the immune response. It's like the immune response goes overboard. So, and lipid A is one of the agents that can cause that kind of clinical shock. The polysaccharide also is antigenic. So here, have a look and you'll see. Here is the lipid A, and that is embedded into the lipid bilayer of the outer membrane. And here, attached to lipid A, there is a polysaccharide molecule, very complex, lengthy polysaccharide molecule. And that in itself is antigenic. But it is this lipid A which is particularly dangerous. And it is called an endotoxin. Now, what happens is in a gram-negative bacterium, if it is in circulation and it dies and bursts and lyses, it will liberate lipid A in solution into the body fluids. And once it enters the body fluid, begins circulating, it excites this very, very strong immune response. Gram-negative bacteria in a pathogenic situation are particularly dangerous because of this. Gram-positive bacteria certainly also have antigenic properties and can induce all sorts of immune response. But gram-negatives can induce this particularly dangerous strong response. Okay, so the condition which results from that, this clinical shock, is called septic shock. And this is a situation that is referred to as sepsis. Sepsis is not just the circulation, you kind of think instinctively sepsis is just circulation of bacteria. It's not. Sepsis is actually a physiological response to the presence of antigen, which produces a catastrophic drop in blood pressure and organ failure. And we will discuss it in more detail later on when we talk about clinical situations. Just let me check that I've told you everything I need to tell you. Oh, yes, this is important as well. These embedded proteins here are transport proteins, and they are called porins. And the porins are specialized transport proteins. This is not active transport, but they are able to recognize and transport very often very rare molecules that are present in the medium around them. And there are many of them in gram-negative bacteria, highly specialized for transporting molecules into the periplasmic space, into the periplasm itself. Okay, so I think that's what all. So you've heard this term gram-positive and gram-negative. What does the gram-positives have? A thick cell wall, I said. Gram-negatives have a thin cell wall and the outer membrane. So the term gram comes from the name of somebody. It was Professor Gram, who developed the staining technique, which is really the most standard clinical staining technique. It's the first thing you do if you have an unknown bacterium in a clinical lab or wherever. You always perform a gram stain, and that immediately gives you an enormous amount of information. So I'll run through how the gram stain is done and then explain why, in actual fact, the reactions are different. Okay, so what we're going to do is we're going to take, this is what you'll do in lab. You'll take a mixture of gram-positive and gram-negative bacteria. A typical gram-negative bacteria is E. coli, which you've heard about often. A typical gram-positive would be any of the cocci, like streptococcus or staphylococcus. And then there are a lot of gram-positive rods as well. But in this case, what we're doing is we've got a gram-negative rod and a gram-positive coccus that we're going to stain. So first of all, you need to be careful when you do this. You make a smear in water, spread it out very, very, very thin. When you do this in lab, make sure that you do this extremely, extremely dilute mixture of the bacterial sample and water. You spread it out, then you let it dry, and then you fix it. You heat fix it. What you do is you heat up the slide, and that causes all the bacterial proteins to coagulate and tie the cells to the glass slide so they don't wash off. The very first thing you do is you flood the slide with a dye called crystal violet. Crystal violet is just a good general dye, and it stains all of the cells, whether they're gram-positive or negative. It stains them all a deep blue color. So we then, you leave that for a requisite amount of time, and then you add iodine. Now, iodine is called a mordant. And what that does is it takes the crystal violet and it ties it very tightly to the cell, and specifically to the peptidoglycan, but also to elements inside the cytoplasm. It's like a way of retarding the crystal violet so it's not easily removed. You then, this is the critical step in the lab. This is an alcohol wash. You then wash the cells with alcohol. Crystal violet is extremely soluble in alcohol, very, very soluble indeed. In the case of the gram-negative bacteria, the crystal violet simply washes straight out. But in the case of the gram-positives, it is retarded inside the cell, and it's principally retarded by the very thick peptidoglycan layer. You have to be careful at this step. This has to be done very briefly, because if you leave the alcohol on too long, you can wash the crystal violet even out of the gram-positive bacteria. The reason that the gram-negative bacteria respond this way is, first of all, they don't have that thick layer of peptidoglycan with the crystal violet mordanted to it. But in second reason is that the alcohol actually disrupts the outer membrane. And when that happens, everything that's trapped in the cytoplasm and this periplasmic space washes out very easily. So the crystal violet is lost from the gram-negative very easily in that step. So now what you're left with, were you to now look at it under the microscope, what you would see, you would actually only see the gram-positive bacteria stained deep blue, kind of deep blue-black actually, color by this stage. And you would see in the background, you might be able to see the kind of ghosts of the gram-negatives, because they will be completely colorless at this stage and difficult to see. So that's not a situation we want. We want to be able to see our gram-negatives easily and distinguish them. And so what we then do is we stain with a general stain called safranin. And safranin will stain all of the cells. But in the case of the gram-positives, you can't actually see the safranin because they're so deeply stained with crystal violet. Instead, it will stain the gram-negatives a pink color like this. And this is what you end up seeing if it all works properly. These are the gram-negative bacteria in the background here, the rods here, and here are the gram-positive cocci, all stained a deep blue, kind of a blue-black color. It's a very simple staining technique, but it's tremendously important because it produces the first step in our ability to classify the bacteria and start to identify them. And there are clinically significant differences between gram-positive and gram-negative. Now, the gram-negative bacteria are regarded as being an evolutionary advance on the gram-positive. That gram-negative outer membrane structure is actually extremely active. It provides protection from all sorts of things. It provides protection from detergents, from antibiotics, which might be coming in, and that sort of thing. But it also allows the gram-negative a special quality of accumulating rare molecules. And so one of the things that we notice is in environmental samples, for example, anywhere in an aquatic situation where nutrients are very rare, for example, in seawater or in lakes or in streams, the bacteria you tend to find are gram-negative because they can exist in that environment, that low nutrient environment. They're much better than gram-positives. Gram-positives tend to occur in nutrient-richer environments. It's not a universal rule, but you tend to find more gram-negatives gram-positives in a rich nutrient environment like the soil, the gut, these sort of things. That is not a universal rule. Many of our gut bacteria, for example, are gram-negative, but we do have many gram-positives in our gut as well. Okay, so we need to talk about just one. There are some exceptions to this gram-positive, gram-negative division. And here's one of them. And this is, we mention this because this is such an important bacteria. This is mycobacteria. And the mycobacteria are a group which are very diverse. Some of them are completely harmless. And in fact, we have mycobacteria which are commensals with us. They exist on our skin in various places. They don't cause any harm at all. They're just part of the natural flora that we harbor. But there are some which are extremely dangerous pathogens. And here's a problem with mycobacterium. They have a specialized cell wall. And you can't use the gram stain. They don't respond to the gram stain. They're very difficult to stain using the gram method. And instead, we have to use a special staining technique. And the reason for this is that the cell wall contains a substance called mycolic acid. And mycolic acid is a wax, and it repels stains which are aqueous, like crystal violet, for example. So instead, we have to use a special staining technique. And what we do is you will do this in lab. We take the bacterium and we actually heat them up. And in heating it, we melt that mycolic wax enough that it allows stain to penetrate to the cell and stain it. You then cool it and you let that mycolic acid set, and you then wash it with acid alcohol. And any cell that does not have that waxy cell wall will liberate all its stain and will not be visible. The mycolic acid, however, seals stain inside the cell if it is present. So in those cells which have mycolic acid, when you acid wash them, they stay stained pink. All of the other cells lose all their coloration, and we then counter stain with a blue stain. So cells which do not contain mycolic acids are now stained blue. The mycolic acid containing cells stain pink, and they repel the blue stain. They can't stain them at all. When we look through, we look at a smear, for example. Here is a smear, probably a sputum, from somebody that they suspect is suffering from one of the diseases, which we'll talk about in a minute. And here we can see these distinct pink rods. If you see here, you see how they are stacked up against one another like that. That's called palisading, and it's another typical reaction of this particular bacterium. In the background here, we've got a whole gamish of mucus and other cells, because this is being coughed up from the lungs. There are epithelial cells and things like that. You can see they are stained. And then if you look carefully, you'll see embedded on them are deep blue stains. Those are bacteria, but they don't contain mycolic acid. Only these pink ones. And we say these pink ones are acid fast. They did not get decolorized with an acid wash, an acid alcohol wash, and instead they kept their dye, the pink dye, so they are acid fast. And this is typical of this genus, Mycobacterium. In Mycobacterium, there are two diseases which are really important. And the first is Mycobacterium tuberculosis, which the name tells you immediately is tuberculosis, principally a disease of the lungs, but tuberculosis may spread through the whole body as well. We'll talk about it much later on because it's a really important disease. The other mycobacterial disease, which is important, is Mycobacterium leprae, caused by Mycobacterium leprae, and that is leprosy. Now it's not politically correct to call it leprosy anymore. Instead, it's called Hansen's disease, because Hansen was the first person to describe it. but they're very important pathogens. And that's a situation in which you would do an acid fast staining. Okay, so having discussed the cell wall, now let's return to our discussion about the cell membrane. We've got a good idea now about the structure of the cell membrane. I would say that many of the things I'm going to talk, many of the principles I'm going to talk about in eukaryotes would apply to the membranes which surround the organelles as well. But of course, in our consideration, we're only talking about prokaryotes. So there is only one membrane to discuss, and that is the cell membrane or the plasma membrane around the outside of the cell. This thin membrane, just 2 molecules thick, is actually a very profound barrier. And the reason for that is this hydrophilic, hydrophobic nature, because most substances dissolved in water or in water are actually hydrophilic. So they find it extremely difficult to move across this barrier because they have to get through this hydrophobic portion in the middle. In addition, anything which is charged, any charged molecule finds it extremely difficult to move, would find it extremely difficult to move through the cell membrane because of interactions with these hydrophilic heads in particular. So this schizophrenic nature of the molecule, of the phospholipids, is what confers the ability to actually exclude molecules and prevent them from moving across. There are some which can get across. Gases can get across quite easily. And surprisingly enough, water itself can move across the membrane, but it does so extremely slowly. The gases can move quite easily. But anything which is charged, ions, all of these things, they find it very difficult to do so. Some of them may drift across, but slowly and in an uncontrolled manner. So those are the principle of diffusion, remember, is that things move from area of high concentration to an area of low concentration. And so that is called simple diffusion. If, for example, I have a high concentration of oxygen on the outside, it will move across the membrane to the area of low concentration on the inside. But most molecules cannot do that. And they would just be unable to get to the inside. Of course, that's an untenable situation for the cell. The cell has to be able to accumulate molecules which are useful to it and necessary in the metabolic processes. So it has to get the stuff across somehow. Now, the first way in which this is done is called facilitated diffusion. It's not simple diffusion. It's facilitated because the cell sets up a situation where molecules can move easily across the membrane, but it does not require energy. So there are 2 ways in which this can be done. The first is through transport proteins, which are not particularly specific. Sometimes they're specific for a particular group kind of molecule. Maybe we've got a transport protein, which allows sugars to go across or something like that. But these are non-specific. They don't limit transport to one particular molecule. So facilitated diffusion always works using the natural forces of diffusion from high concentration to low concentration. And we may find it in both directions, by the way, but most commonly it's to move molecules into the cytoplasm from the outside. So there's non-specific transport like that. The cell can exert some control over that, however, simply by removing the transporter if it's no longer needed. So it may, when it needs to, it can manufacture lots of these proteins and shove them in the membrane to facilitate diffusion rapidly. No longer necessary, they can pull them out and shut them down. But there are also many specific transporters. So, for example, there are specific transporters for glucose. And these usually work by enabling a reaction between the substance that they're going to transport, let's say glucose. Glucose actually binds to the protein of the transporter. And in binding here, it alters the structure of the protein. And the protein now opens to the inside. And once it opens to the inside in that particular conformation, it loses its ability to bind the substance it's transporting, and it releases it to the inside. So we have glucose, it comes to the glucose transporter, it binds to the protein, it alters its structure, and the transporter loses its ability to bind the substance, and it releases it. So it can actually very, very rapidly, this can happen thousands of times a second, it can very rapidly transport the substance across the membrane. But this requires no energy. It is a passive process. It's a facilitated diffusion, but it is a passive process. Now, a kind of specialized version of this is that the cell can also transport water across the membrane. I told you can get some diffusion of water across the membrane. It is very slow, and far too slow very often for the cell's needs. And instead, when the cell needs to transport water, it will open up these specialized transporters called the aquaporins. And the cell can actually manufacture aquaporins as needed, embed them in the membrane, and allow the very rapid transport of water into the cell. You'll realize from what I've told you before that getting water into the cell is seldom a problem. And the reason for that is that these aquaporins are embedded in the membrane. There is some control over it simply by limiting the number of aquaporins in the membrane. But there is, once the aquaporin is there, it passively transports water across from the outside to the inside. Okay, so that's here are different ways of transporting molecules to and from across the molecule. First of all, by simple diffusion. That is limited to only a few kinds of molecules. Next, facilitated diffusion, either through non-specific transporters or else by specific transporters which respond to the presence of a particular molecule, such as glucose. All of these are passive transport systems. They require no input of energy. But very often, the cell needs to move substances across the membrane from an area of low concentration to an area of high concentration. Now, that's the opposite. That is going against the diffusive nature of these molecules. And in order to do that, the cell has to put in energy. And this here is a typical, this is a typical and very famous one. This is a potassium sodium pump, which occurs in bacteria, occurs in eukaryotes, very, very important in nerve cells, for example. And what this is doing is, first of all, let's just do it this way around. We're going to take this molecule here. You can see that it is rare on this side here. It's actually potassium. And we're going to bind it here to these here. And when we do so, this molecule loses its ability to bind these square molecules here, and they are released. Energy is then used to change the formation of the molecule, and the transport molecule opens up towards the outside like this, loses its ability to bind these potassium molecules and releases them. When it's facing out like this, it no longer can bind the potassium, but it will bind the sodium. It binds the sodiums to the protein, and the molecule changes shape and faces inward. Now we repeat the whole process. We have to put in energy. That when we do so, it releases the sodiums to the inside. It changes shape, and it releases the potassiums to the outside. So it's pumping in two directions, but it is specific for two different molecules. Pumps one to the outside, one to the inside, one to the outside, one to the inside. And as it does so, it burns energy. It has to burn energy in order to do these changes in shape. The energy that it uses is derived from ATP, the most important biologically useful form of energy. ATP is converted to ADP. ATP is adenosine triphosphate. It is converted to ADP, and the energy contained in that last phosphate bond is used to perform this transport back and forth. Okay, so let me go on now to, let me just say something. Please make sure that you go over this structure and functions of the membrane very carefully in your book, because you are going to be eventually be closely examined on those topics, okay? And it's really important that you understand how that works. Okay, so let's return to our general cell anatomy and have a look quickly at some of the cytoplasmic elements. We've talked a lot about the outside of the cell, but now let's talk more about the inside of the cell and some of the things that are associated with the inside of the cell. Bacterial chromosome is one thing I've discussed in detail before. A very large DNA molecule in the form of a circle, which is intricately wound up in an area of the cytoplasm called the nucleoid. So that is the bulk of the DNA in the cell. It is not all the DNA, because many bacterial cells will have little circlets of DNA called the plasmids, also in the cytoplasm and containing very often useful information, such as antibiotic resistance. Sometimes it confers the ability to produce toxins. Sometimes it confers the ability for a non-pathogenic cell to become a pathogenic cell. We'll talk about one of those in a minute. Okay, so that I'm not going to say much more about. We discussed it in detail before. But also inside the cell, you may see many other things. Once you develop the ability to examine these cells carefully, you'll start to recognize that there are maybe things inside the cytoplasm which sometimes mimic, visually mimic the organelles, but they're not organelles. They're never membrane bound. And these are referred to as the inclusions, and inclusions may be many different kinds. Very often they are storage products. They store phosphate compounds, they store polysaccharides, especially glycogen. That is one that's commonly seen. And in this case, they're storing this substance, and we abbreviate it to PBH, polybeta hydroxybutyrate. And here you can see there's large stores of it inside the cytoplasm. That's actually very useful. So the cell doesn't need to use all its pH at one time, so it can store it for future use. You'll see many different substances stored like this inside the cytoplasm. But the other inclusion that you'll see is one which is restricted to two particular genera of bacteria. And these are restricted to Bacillus and Clostridium. We'll hear a lot about both of these later on when we talk about bacteria and disease. But Bacillus is a very common genus, and it is a facultative anaerobe. That means it can operate in the presence of oxygen, or it can operate in the absence of oxygen. It's a very common environmental bacterium, but there are situations where it causes disease. And there are some very important diseases, one of which we've already talked about, which is anthrax, caused by Bacillus anthracis. The other genus that performs this activity is Clostridium. And Clostridium, these are obligate anaerobes. Obligate anaerobes cannot tolerate the presence of oxygen at all and only operate in the absence of oxygen. So these two genera are able to produce spores. Now, spore, in this case, is a specialized cell. This is not a way of reproducing. This is a production of a specialized cell. Here is the normal, you can think of, think of that there, that portion as being a normal cell. That we refer to as a vegetative cell. It's out in the world and it's operating with all the normal things. It's growing, it's sopping up nutrients and dividing and reproducing and everything else. But normally when it reproduces by binary fission, and it produces two identical cells which are clones of one another. But under special circumstances, especially when conditions begin to get bad in its growing environment, when nutrient becomes low or when waste products start to build up, then the cell responds with a special kind of division. And it will divide so that here it replicates its DNA, shunts one molecule of DNA to an area of the cell where a small cell begins to form. And the membrane of the cell pinches off and actually isolates this area here. We call this a prospore. And we then have a process whereby another membrane is laid down around it, and a layer of peptidoglycan is laid down between those two membranes. Now this outside of this membrane on the outside, a thick wall is laid down. This wall is highly specialized, and it's called the spore wall. And now the spore actually begins to be quite visible, even in an unstained state. You can often see the spore quite clearly. And this spore wall here, we'll have a look here at this electron micrograph. What we see here is the inside of the cell, what would be the cytoplasm here. There is a membrane around this. Here's another membrane. The black line here is the other membrane laid around the outside. And between them is the peptidoglycan layer there. But the really important feature of the cell is this. All of this here is the spore wall, a very thick layer of complex proteins and polysaccharides. And that spore wall confers on the spore all sorts of abilities. First of all, the spore becomes almost entirely dormant. There's almost no metabolism at all inside the spore. So it's in a resting state. The spore wall allows the spore to be resistant to antibiotics, to detergents, to alcohol, even to boiling. It can withstand boiling. And if we want to kill spores, we actually have to raise the temperature above boiling point. And the only way to do that is to put whatever we want to sterilize, or the spores we want to kill, into a pressurized cylinder, into a pressurized vessel, where we raise the pressure and then boil water, because water under pressure will boil at a much higher temperature. And we can raise the temperature up enough under those circumstances to kill spores. And that apparatus that we use, the pressure cooker we use to do that in, is called an autoclave. And all of the media and everything that you use in surgery, all of the stuff that is used in surgery, this is all sterilized, not only to remove living bacteria, but especially to get rid of spores. And because both of these, Bacillus and Clostridium, have members which are highly pathogenic. And if their spores are present, once the spores land in a suitable environment, a wound, for example, or an aqueous solution that is suitable for their growth, the spores will germinate and form new bacterial cells and rapidly reproduce. The process of producing the spore is called sporulation, and that's this whole process here, budding off a small portion of the cell, laying down membranes, peptidoglycan, and then a thick layer spore wall on the outside. Okay, so there are, in fact, even more features on the outside of the cell wall itself. We saw, for example, we have the cell membrane and the cell wall. There may be features outside the cell wall, and the most important of them is a slimy layer of mucus. Now that can have different natures, depending first of all upon the organism, but second of all upon the conditions under which the organism is living. If it is, the slime is like mucus, it's a mixture of all of proteins and polysaccharides like mucus. If it is very well organized and easily visible, we call it a capsule. And this is an important clinical feature, as I'll explain in a minute. Here you can see here, this has been a special stain. Here the cells are stained dark, but the background of the preparation is stained. And this clear portion here is the capsule. And the capsule is excluding this background or negative stain. The cells that produce the capsule are stained here with another stain. But here we can clearly see, this is you'll do this in lab, I think, and you usually do this just with Indian ink. And the capsule repels the Indian ink and leaves this clear zone. Now, sometimes this the outer layer is actually loosely organized and can actually wash off quite easily, and that we call a slime layer. Now, these layers are important because of several things. First of all, they are protective. They do, but they especially protect against the activity of the immune system, for example. They protect against phagocytosis by white blood cells. Second of all, it's these layers that allow cells to begin to aggregate together. Bacteria love to grow in a layer, and they love to grow on a surface. And these, either the capsule or the slime layer, allow them to do that much more easily. But there is another aspect to this. The capsule is especially important for attachment. And attachment is often the first stage of pathogenesis. So there is one particular instance where this is really famously been documented in molecular biology. And this is in the case of a coccus which causes pneumonia. And this Streptococcus pneumoniae, which causes pneumonia, but it occurs in two forms. There's one form which has a capsule like this. There is another form which lacks the capsule. And when you grow them on an agar, the one that grows the capsule looks smooth. The colonies look smooth because of all the mucus. The ones which lack a capsule look rough. And so they are often called the rough strain or the smooth strain. And what is interesting is that the smooth strain is pathogenic. The rough strain is not pathogenic. And the reason for that is that the smooth strain has a capsule, and the capsule allows the coccus to bind to the lung cells and reproduce and start to produce a pneumonia. And this is often a very virulent pneumonia, especially in people who are bedridden or the elderly. The rough strain cannot do that. It cannot bind to the cells, and it can't start the infectious process. Why this is so famous is because somebody, a scientist demonstrated that the ability to produce a capsule could be transferred. It could be transferred from a smooth strain to a rough strain, and the rough strain would then become smooth. And this was the very first demonstration that DNA can actually be transferred from one bacterium to another, quite apart from all sorts of other things that were important about that experiment. That we'll discuss when we'll talk more about molecular biology. Okay, so here's a capsule. This is a slime layer, loosely organized around. And these capsules and slime layers can occur in gram-positive and gram-negative bacteria. It's not associated with one of them. Another thing which occurs on the outside of cells are the flagella, and these may be clinically significant for various reasons. First of all, we use the flagellation of cells, how their flagella are organized as to characterize the bacterium. But second of all, flagella in pathogenic bacteria are antigenic. So the immune system responds strongly to the presence of flagella. Bacteria may be completely absent of flagella. There are bacteria which simply don't grow flagella. In those which do, the flagella can be produced extremely quickly. Sometimes you'll see that the cells do not have flagella, and then you put them into another situation, and they will actually produce flagella. And flagella, how they are flagellated is characteristic and genetically determined. So, for example, here is one. This is very common. This is a very common situation. Here's our bacterial cell, and it has flagella all over the cell. And that's called peritrichous flagellation. Sometimes they have only one flagellum, and here this is monotrichous flagellation. A lophotrichous flagellation is where you have a whole bunch coming out from one place. Well, this is lophotrichous. And amphitrichous is when you have flagella at both ends, both there and there. So, and this is called, these are amphitrichous flagella. So the flagellum is, I'm not going to go into it in a huge amount of detail, but it's a very fascinating part of the cell because of its incredible organization. Here you can see here, it has actually a little motor embedded into the cell membrane. This is a gram-positive, but in gram-negative, it has two sets of rings, one embedded in the outer membrane and one embedded on the cell membrane. And these are driven, the motor is driven by ATP. It uses, and it actually uses a lot of energy to drive the motor. And the motor can spin in both directions. But there's only one direction where it is functional and will produce movement. Now, it's tempting when you see the shape of the flagellum to think that the flagellum does this, right? It doesn't. The flagellum is actually a rigid, more rigid helix in space, and it spins like a propeller. So, and it's spun by this little motor, and from here, this is called the hook, but it goes on from there into the flagellum itself. So the way in which this works is that there are two modes of movement with the flagellum. There is a useful rotation, in one direction. And when the flagella are spun in that direction, they get organized and they spin as a unit and move the cell, and they'll move it almost straight ahead. And that is called a run. So here you can see, here the flagella are all beating in this direction, and the bacterium will move in a useful direction. Now, let's just say I'm going to leave my pointer here. I mean, just imagine that I'm leaving my pointer here in the center, and the bacterium is trying to get to this place here. So it's running in this direction because it's picking up molecules or whatever that are in high concentration here. It's moving towards them in an organized fashion, but soon it's going to start moving away. Now, the bacterium has sensors that are picking up information from the environment. So it picks up that the signal it wants is fading. And when that happens, the flagella switch and they beat in the opposite direction. And when that happens, they become disorganized. And the cell just begins to tumble randomly. And it will tumble randomly until it picks up the signal again. And it will then start a run. And by doing this, by doing alternating run and tumble, it will slowly begin to move towards and finally home in on the attractive signal. Now, it can do the opposite. It can also move away from noxious substances or a bad situation. It can move away from a high carbon dioxide area to a high oxygen level, for example, or it can move away from a toxin being produced by something and away into safety. So although it sounds like an inefficient way of operating, this run and tumble, nonetheless, it functions for the bacteria. There are lots of kinds of bacterial movement, I should tell you. This we have mentioned before. This is a spirochete. And a spirochete, for example, Leptospira is one of them. And Treponema is another one that I told you about. They're organized somewhat differently. They have this rigid corkscrew-shaped cell. And wrapped around that corkscrew are these, they call the axial filaments, but they're actually modified flagella. And when they beat, instead of beating in the medium outside, they now force this corkscrew-shaped cell to start spinning on its own axis and moving forward. And the cell is acting as its own propeller. There's a specific reason why we think this evolved in these organisms, and that is that it allows them to burrow. It allows pathogenic spirochetes, for example, to burrow through mucus layers, which are normally protective of the mucous membranes. Environmentally, it allows spirochetes to burrow into the medium. It allows them to burrow through material, like or whatever is leaves or whatever are decaying in water, for example. So it's a very useful way of moving it, and it actually allows the cells also to move backwards. And spirochetes are the only bacteria which can move forward and back. The others are restricted to run and tumble, but spirochetes can move forward and back. Here's another set of extensions from on the outside of the cell. And these are different hair-like structures. These are made of protein. We don't worry about the difference between fimbriae and, this is pronounced pili. Fimbriae and pili. There are various functions associated with these extensions. One of them is to confer strange forms of movement called twitching and gliding. Sometimes you will see bacteria which can actually glide along surface. In other cases, they move by a twitching motion, which flings them up into the medium, and they then land and twitch again, like jumping beans. But there's one type of pilus, which is extremely important in the transfer of DNA between cells. And this is a long pilus, a very large one, which looks more or less like a straw. And here it is here extending between two cells. We will hear more about this when we talk about molecular biology, DNA, and the function of DNA. But here's an example of it. Here is a cell which has extended a pilus. It is called a sex pilus, even though this is not really sexual reproduction, but it is the exchange of genetic information. What happens is not that the cell spits the DNA all the way down this tube. Once the sex pilus is established, it shortens and it pulls the one cell close up against the other. And then where the sex pilus was, an opening is made between the two cell membranes. So the cell membrane, the cytoplasm of one cell is now in contact with the other. And through that opening, DNA is transferred in a specialized manner, which we'll discuss much later. But just remember, the sex pilus is for the transfer of DNA from one cell to another. Okay, so that's the end of our discussion about the form and function of prokaryote cells. I do want you, please, to revise all the material that you had about the organelles and structure of eukaryote cells. This very beautiful photograph, by the way, is a specialized microscopic technique called confocal microscopy, which produces these beautiful, very, very clear pictures. And in this case, I'll just point out, the blue is a nucleus. This is a cell, actually, here. This whole thing there is a cell. And what is stained green here are parts of the cytoskeleton of the cell, which maintains the structure of the cell, but may also be involved in transport. There's very beautiful pictures. But please make sure that you revise and understand structures of the eukaryote cells, and especially the differences between eukaryotes and prokaryotes, and then the differences between animal eukaryotes and plant eukaryotes. And that's it for the